Human embryogenesis is a process of the development of the human embryo. It has some phases: fertilization, cleavage, gastrulation, cystogenesis. Let's start from the first one. Fertilization. Fertilization is a process by which the male and female gametes fuse, makes the beginning of the pregnancy. Fertilization is the fusion of the genetic material of two sex cells, male and female, with formation of the unicellular organism, which is a zygote, unicellular, and the diploid organism, zygote. It has two phases: distant phase and the contact phase. Before meeting sperm cells and oocyte, sperm cells pass through female reproductive phase and undergo influence of the mucus of the vagina and get and finish maturation. And all of substances which cover head of the apical anterior part of the head of the sperm cells is slowed by the enzymes of the mucus of vagina. It's very helpful for movement and fluid penetration and fluid meeting and fluid interaction between the sperm cell and the secondary oocyte. And this first subphase of the distant stage is named capacitation. Next phase is a contact. Contact phase is beginning since the moment of being a sperm cell very close to the secondary oocyte. The contact stage, the first sub phase of the contact state is the acrosomal reaction. What does it mean? Acrosomal reaction process of liberation of enzymes of the acrosome for the mating of layers of the secondary oocyte which surround it, like zona pellucida and corona radiata. Let me remind you that acrosome, it's a special structure on the anterior part of the head of sperm cell, which is a modified Golgi apparatus which contain vesicles rich enzymes such as hyaluronidase and proteinase, which very helpful for penetration of outer layer of the secondary oocyte, which are corona radiata and zona pellucida. And first of all, for penetration of layers, for getting to the cytoplasm of the secondary oocytes, sperm cells must release proteinase. And after the maiden of the corona radiata, the next layer would undergo the maiden. It's the zona pellucida by the hyaluronidase, because zona pellucida mainly is composed by hyaluronic acid. It's a cellular structure and which undergo influence of the hyaluronidase and must be penetrated by the sperm cell. So during the acrosomal reaction, corona radiata, zona pellucida, and plasma lemma of the ovum must be penetrated, and also the sperm cell loses the teeth. So pay attention that during fertilization, secondary oocyte finish second phase of the meiosis and became the ovum. After acrosomal reaction, the cortical reaction goes. It means after penetration of the plasmalemma, happen liberation of cortical granules which are located before it in the peripheral part of the cytoplasm of the ovum, and after penetration they leave peripheral part of the cytoplasm and take place in the perivitellin space, which is located between the plasmalemma of the ovum and the zona pellucida, and create unpenetrable layer which prevent polyspermia, so that it's impossible penetration of the layers of the ovum and the plasmalemma by other sperm cells. The human fertilization happens in the ampulla part of the uterine tube. The process of next development of the zygote is followed by movement of the zygote toward the space of the uterus. The next phase of the human embryogenesis is a cleavage. The cleavage is a set of mitotic divisions which go one by one, which result is the formation from the unicellular organism, which is a zygote, multicellular organism. The first kind of multicellular organism is a morula. Special properties of these divisions, which are named fragmentation or segmentation, is that the next generation cell of cells became smaller and smaller and smaller, and this mitotic division followed by decreasing of size of cells, increasing of number of cells. This happened because of short interface between mitotic divisions. Here you can see morula. Morula, it's a multicellular organism which is a result of some numbers of mitotic divisions during cleavage. The human cleavage, by the classification, is a full, subequal, and asynchronic. Morula undergo next cleavage and became blastula. Blastula is a multicellular organism which contains inside space a blastocele, and the wall of this space is named blastoderm. Human type of blastula, it's a blastocyst because of shape. As a result of unequal cleavage, we have formation of two types of cells: inner mass of large duct cells, which will give rise to the embryo body, and outer cells, which are small and light cells, which will give rise to formation of the trophoblast. On the 5th day after fertilization, on the stage of the blastocyst, the embryo entered to the uteral space, where since 6th till 7th day happen implantation. And distinguish two phases of implantation: adhesion and invasion. Adhesion, adhesion is attachment of surface of the blastocyst to the surface of the endometrium. We see next relation of enzymes which are produced by trophoblast, and the maiden of the surface of the endometrium with formation of the fossa for embedding of the blastocyst deeply inside into the endometrium, and this phase of embedding is named invasion. Trophoblast at the moment is composed by two layers which are syncytiotrophoblast and cytotrophoblast. Syncytiotrophoblast, cells which compose syncytiotrophoblast produce special enzymes which break the surface of the endometrium and very helpful for invasion stage of the implantation. The next stage after cleavage is gastrulation. Gastrulation is a compound process of chemical and morphological changes in the embryo, which leads to formation of derm layers, ectoderm, endoderm, and mesoderm, and the formation of axial organs. Human gastrulation past next stages, the delamination and immigration. At the first, in the embryoblast of the blastocyst, there happen delamination. It's the division of the embryoblast to two laminas. As a result, the formation of two layers, upper layer epiblast and lower layer hypoblast. The cells of the epiblast undergo division and go up with formation of the amniotic cavity, and the margins of the hypoblast during this process go down with formation of the yolk sac. The bottom of the amniotic cavity and the roof of the yolk sac will give formation of the embryonic disc. Cells of the bottom of the amniotic cavity begin migration. As a result of this migration, you can see formation of a special pattern. The special aggregation of cells. It's a primitive streak and primitive node on the anterior ending of this primitive streak. In the center of this primitive node will organize the fossa. It's a vagination, it's a primitive fossa. In the center of the primitive streak will organize the primitive growth. Here is a picture of the formation of the embryonic disc, and all of these images is a result of migration of cells. During migration, cells of the epiblast migrate through the layer of the epiblast take place in the space between epiblast and hypoblast with formation of the middle layer, which will give rise to the mesoderm. Epiblast will become ectoderm and hypoblast will become endoderm. After formation of the mesoderm as a middle layer between ectoderm and endoderm, it undergoes differentiation to paraaxial mesoderm, visceral, intermediate mesoderm, and ventral mesoderm. Paraaxial mesoderm is close to the middle part of the body, to the axis of the body, undergo segmentation to somites. And each somite is segmented to three parts: myotome, dermatome, and sclerotome. Myotome will give rise to the skeletal muscle tissue. Sclerotome to the connective tissue with the cartilage and bone tissue, and the dermatome to the dermis of the skin. Intermediate part of the mesoderm will give rise to the urinary and urogenital system, and the ventral mesoderm will be divided to two layers: parietal and visceral layer, and between them take place coelomic space. Since 14th till 17th days after fertilization, happen formation of axial organs: notochord, neural tube, and gut tube. Notochord organized from the cells from the material from a primitive node. Neural tube, notochord is here. Neural tube is organized from the ectoderm. In the middle part of the ectoderm will happen vagination evagination of the central part of the ectoderm, this formation of the first neural plate, neural groove, neural tube, and in the future will give rise to the central nervous system. After that, all the ectoderm became skin ectoderm. The gut tube organized from the endoderm. And the part of cells we don't give formation of the neural tube, but which are cells of the neuroectoderm, will give formation of aggregation from both sides of the neural tube, which are named neural crests. Neural crest and cells of the neural crest will give formation, for example, of spinal ganglia and the Aput system cells. And the dilation portion of the ectoderm in the place of the future head will get name neural placoid. Process of formation of axial organs in the embryo is followed by differentiation of the trophoblast. At the first, it's the two layers, which are cytotrophoblast and syncytial trophoblast. Cells of the cytotrophoblast divide by mitosis with formation of villi. Actually, we have formation of three types of villi: the primary, secondary, and tertiary villi. And the future, they will give rise to placenta, which is a fetal organ. Except placenta, the embryo has the next fetal organs, such as umbilical cord, allantois, yolk sac, and amnion.